So next we will go for some global alignment with the help of fast alignment tool. So we will go for fast alignment tool now. FASTA. So FASTA alignment tool is available with the EBI, European Bioinformatics Institute. You can see now here. So we go for this FASTA alignment tool. We can also use the same sequence. So the difference between BLAST and FASTA is a global alignment. Global alignment means it will compare the whole sequence to the sequence available in the database. Whereas BLAST is a local alignment. It will compare only part of the sequence. Okay. So now this is the FASTA tool. Here we will submit our sequence of interest. And then its sequence is protein. So we leave it as a protein. And we are going to use the program FASTA. And then we can give any title, similarity studies. Okay, then you can submit. Similarly, you can change the parameters as shown in the BLAST also. Here you can say you can change the matrix, you can change the gap open value, gap extension value, k-tuple, word size. In BLAST you can see the word size. Here instead of the word size, it is called as a k-tuple value. So what is the fragment which can be used for comparison? So like this, you can change many parameters of this FASTA tool. If you want, you can change. Otherwise, you can leave it as default values. Then you can submit. So once you submit it, it is actually queued. Then it has to be run. Okay, now it is running. So once it is completed, you can get the result. So now you can see now, this is the FASTA output. Here also you can see the sequence compared with other sequence which is available in the database. You can see this percentage of identity of our sequence of interest with the human osteopontin. Here mus musculus osteopontin is matched with 76% similarity. This organism is also going to have 85% of similarity, and you can see the other organisms also listed here. Okay, so Bostaurus is having 74%, 74.5% similarity with our sequence. You can see here identity. Identity means both the sequence are identical. Similarity means slight change is also accepted. That is what the difference between identity and similarity. That is positivity, it is mentioned here. So you can see different sequences which are similar to our protein of interest are represented in the tabular form. So now we can identify our sequence of interest similar with what are the other sequence. You can see here the organism wise list. So there are about, our sequence of interest is related with 4 human sequences. Rats we can see about 3, Bostaurus you can see 3, mus musculus we will see about 3 sequences, and other organisms also listed here. So we can identify that what are the other sequences which are having similarity to our sequence of interest. Again, if you want to see the visual output or the tool output, the similarities, we can see here the list, how the sequence alignment. Now, like in the BLAST tool, here also you can see the alignment. Here you can see the complete alignment of the whole sequence. See, this is our sequence of interest, and this is the sequence available from the database. So from our sequence of interest, you can see right from the first base to the end of 300 base pair, you can see the comparison. Whereas in the case of NCBI, if you go for an alignment, you will see only mostly the part. You can see now the query start with the 15th base. Okay. Similarly, it may end 300 or it may end before that. But in FASTA, almost all the queries will start from first base, and then it will end with the complete sequencing. So that is what the difference between BLAST and FASTA, because FASTA is a global alignment and BLAST is a local alignment. That is the difference. So this is what the output of FASTA alignments. So this is the list of tabular column. So which shows what are the different organisms which are having similar to our sequence of interest. So this is a second part. So local alignment, it is a global alignment,